start.
Okay, we can start. Thank you. Hello, my name is Diana Garcia Bernet. I am a research engineer at INRAE, the French National Research Institute for Agriculture, Food and Environment. I hope you and your families have not been affected by the pandemic crisis that forced us to suspend our workshop in the frame of the BIOKET event in last March. I am very happy that today our workshop, Phenolic acid from research to market, can finally be held. I thank you for attending. I am sure that our discussion will be very interesting and that it would be the starting point for fruitful collaborations. Before we start our webinar, I invite you to hear a message from BIOKET and the IAR poll. Johan, the floor is yours.
Okay, thank you, Diana. Thank you for the kind introduction. While people are still entering in the room, I will start with the introduction word from BIOKET Digital Edition, starting sharing my screen. Okay, perfect. Yes, it's almost time now to start our second webinar. We wait still a few minutes before starting and letting people to enter into the room. And it's time now for some introductory discourse for BIOKET Digital Edition. Today, on paper, registered participants, we are normally more than 200. We will see if everybody can make it to connect and to follow our nice webinar today. Well, ladies and gentlemen, I'm sorry for those who were already here yesterday. I'm going to repeat a little bit the introduction I made yesterday. It is indeed with a very shared feeling that I'm introducing today's program, a feeling of happiness because almost 2 years of work. And finally, here we are delivering the fruit of our work, or at least part of it. Happiness, considering the number of participants or the number of registered people, over 200 every day for a total of more than 700 registered over the 3 days. It is a real success and we thank you for that and we hope to be at the level you are expecting. But also some bitterness and sadness because the sanitary crisis we are experiencing now has upset our entire calendar. And even if we cannot do anything about it, we feel very sorry. We have tried to do our best to adapt and to show a maximum of agility to satisfy our sponsors and you. We appreciate for the understanding of those who have been with us since the beginning, and more in particular, our sponsors. Finally, I want to pay tribute to the whole team of IAR who worked hard on BIOKET to make it happen. Ladies and gentlemen, good morning. My name is Johan de Koning from IAR, the French Bioeconomy Cluster, and welcome to BIOKET Digital Edition. Before starting the program, a few words for our sponsors. We thank them all. They are indeed making a considerable effort, and without them, we would most certainly not be here this morning. Few words for IAR, for those who don't know us.
Let's find out the 7.
Sorry, so I'm always going too fast on that. Okay, sorry for that.
IAR is the French Bioeconomy Cluster. Let's find out the 7 services we provide you to support your innovations. Tomorrow's solutions are being developed today, from ideas to endorsement and funding of your projects. Our expertise and strategic support will be there to maximize your success. Do you want to diversify your activity to reach a new market? Through our research and consultancy services, we analyze, explain, and help de-risk your business and innovation environment, enabling you to make strategic decisions and identify early adopters and clients. Bioeconomy is a European priority. Having been in Brussels since 2008, we can make sure your voice is heard. We are your ideal partner to access European funding. Innovative startup? Our Investors Club is seeking projects like yours. Thanks to our help, reach out to venture funds and raise the necessary funds to boost your project. Speed up your growth and overcome technological hurdles, thanks to the innovation platforms of our ecosystem. Our Bioeconomy Leaders Network offers you unique business opportunities. Meet your future business partners and develop your expertise at the many events we organize throughout the year. Conferences, business conventions, tech days, so many opportunities to increase your visibility and promote your activities. Grow your business abroad and give your ambitions new horizons thanks to our international missions and delegations. Brazil, USA, Canada, India, Southeast Asia, we will enhance your tech and business partnerships. In a changing world, we will help you adapt your training courses to the needs of bioeconomy. Access tailor-made skills, from the endorsement of your training classes to posting jobs. IAR services are here for you. Find out more on en.iar-pole.com.
Two years ago, we were in Strasbourg in the region Grand Est. This year, we were supposed to be in Lille, and we thank our main support, the Regional Council of Hauts-de-France, and we hope to be there next year. We have as well some industrial sponsors, and our platinum ones are the Genopole in Evry, a leading biotechnology platform in France, quite discreet but very effective. They are active in the medtech sector, but also industrial and plant biotechnologies. There is where companies like Global Bioenergy, Insect, Abolis, Innovafit, amongst others, are located. The second platinum sponsor is University of Lille, a major university in France with more than 72,000 students, ranking in top 30 of the most innovative EU university. And we like in particular to thank Frank Duménil from the UCCS, a leading authority in the world of catalysis, with in particular his high throughput screening platform, RealCat, unique in the world of catalysis. We have gold sponsors as well, among which we find our 3 innovation tours. The first one proposed by the Development Agency of Dunkirk, with the Port of Dunkirk, and more specifically Dodouy van Korenberg, a specialist in vegetable recycling, oleochemistry, and biofuel, together with Biotifuel, the Total's pilot unit producing from lignocellulosic biomass via Fischer-Tropsch process biodiesel. The second innovation tour was proposed by Lanolin Stella, a subsidiary of the group De Wavrin, which also includes Isis Pharma and Alpol. Lanolin Stella is one of the world leaders in production of lanolin. They are eager to show you their new molecular distillation unit and installation at your service for tailor-made production. Finally, the third innovation tool was proposed by Pivot, a platform to industrialize bio-based processes, technology, and products. They have everything to host the projects of your choice and to industrialize them. The other gold sponsors are ValueMag, the webinar of yesterday, Tournaire, Optimus, and Tami Silva, 3 engineering companies at your service for equipment and processes dedicated to green extraction and purification, P&O, a leading consulting company for R&D projects, Axens, French technology transfer company, which market many processes to transform biomass. They have an impressive portfolio of technologies. And finally, the last but not least, ARD, no need to present them. They are so much known in the field of process scaling up, in particular in industrial biotechnology with their 220 cubic meter fermenter, a size which allows them to make industrial production to order. Their industrial culture and experience in both biotechnology, green chemistry, makes them the ideal partner to industrialize your processes. We have some silver sponsors, Rodian Novacep, specialist in purification, Noveal, the new name of the industrial subsidiary of L'Oréal, and Vito, a Flemish research organization in the area of clean tech and sustainable development, known for the H2020 BioHorizon project and its LignoValue pilot unit. Finally, we like to thank all our bronze sponsors, and in particular the Institute Carnot Tribeca for the webinar that will follow now. I won't mention them all for fear of forgetting one, but the support of all our sponsors is pretty much appreciated. And you can discover all of them in the slide that is on the screen now. They are as well on our website, and feel free to contact us if you want to enter into contact with them. It will be a pleasure for us to make the matchmaking. Now it is time to switch from the program of today, the webinar. We will still be together for another 2 days with 2 webinars on the program. In a few minutes, we start with the webinar of Carnot Institute Tribeca, a webinar dedicated to phenolic acid. Tomorrow afternoon from 2 p.m. to 5:30 p.m. we finish with the BISBA, a webinar on biofoundries and bioprocesses. We will be immersed in the heart of synthetic biology with the best actor in Europe and in the world. Finally, this webinar will be completed by a networking opportunity and business opportunity with a B2B. There is still 10 hours remaining of the virtual B2B. The platform is still open. You can still register for the remaining 2 sessions and complete your meeting and schedule. You have all the procedure on the screen. Should you encounter any problem, please contact Annabelle. She will be pleased to help you to continue with the B2B. Finally, we are happy to announce a few coming interesting events for the bioeconomy. Next week, in the scope of our intercluster 3BI, we organize a free live webinar dedicated to post-COVID opportunities for the bioeconomy. We have 3 invited speakers, one from Vereniging Nederlandse Petroleum Industry, one from Neste, and one from SABIC. A very nice program to see what are the opportunities that the actual crisis can bring to the bioeconomy. We continue in September with a new BIOKET Digital. We will be presenting another H 2020 project, the project Enabling BBP, a project encouraging the creation of efficient and structured biomass supply chain for the production of bio-based products. Not to forget dates for this fall for other events. They are all digital. I'm talking about the World Bioeconomy Forum on September 10 and EFIB on September 5 and 6. Finally, we are pleased to save the date for our event dedicated to bio-based solution. The Plant-Based Summit is back next June in Reims 2021. We have a brand new advisory board. An original and much more international program is under construction this year, and we invite you all to submit your abstract. The call for paper is already open. But if you should just select one event next year, don't forget the most important one. We hope you are not going to miss it, and we count on all of you to be in March 2021 in Lille. Well, ladies and gentlemen, now it's time to start the webinar. We are very happy to welcome Diana on the floor. Good morning, Diana. Welcome to BIOKET Digital. Happy to finally being able to deliver the webinar. The antenna is yours. I will stop sharing my screen and I will let you start your webinar. Welcome on the floor, okay. Diana.
So you see my screen?
Not yet.
Sorry, I see why. I don't understand why I cannot. Sorry. I don't understand. Sorry. Why I cannot share my screen?
You have the green button on the
yes, no, but I don't see the green button. Sorry, I don't know what happens. Maybe
change the screen.
Sorry. Okay.
Okay, now it's coming.
You see now. Sorry. I'm sorry. I was okay. Okay, thank you. Sorry for this little problem. And thank you for joining us for our today's webinar, phenolic acids from research to market, brought to you by the BIOKET Network, that it will be introduced later, to discuss about scale-up challenges in plant-based chemistry with phenolic acid as an example to illustrate it. Before I introduce our today's moderators, I would like to remind you that our webinar is being recorded, and a link will be shared with you at the end of the webinar. The slides will also be available at the BIOKET Network website. Today, to share this webinar with me, we have Coraline Collet, which is business developer at the BIOKET Network. We have Hélène Roux de Balman, senior scientist from the French National Center for Scientific Research, working at the Chemical Engineering Laboratory in Toulouse, and Jacques-Yvan Deput, scientific director from the IAR Pôle, the French Bioeconomy Cluster. So let's take a look to our busy agenda for this morning. We are going to start with a very brief introduction to the webinar and to the INRAE and the BIOKET Network, and then we are going to start the presentation given by research and development experts. We are going to take as a guiding thread the value chain. So I let you discover in your screen the name of our participants, but the moderators will introduce them in the next session. Concerning the roundtable session, we are very lucky today because we can have the feedback of some industrial and experts dealing with phenolic acid at the different development levels, using different applications, going from the startup case for oleoinol until the well-established market from Solvay and Cepic. Okay, so Coraline,
yeah,
you have the you can start the presentation of the BIOKET Network.
Yeah.
Okay.
Thank you, Dania, for this quick introduction. So I will share my screen. Yeah. So just a few words about Carnot 3B Core. Just to say before, Carnot, it's a French label which is deserved to research structure for their ability to make research for industrial partners, and in a way to offer an already support to these private partners. There is 39 Carnot in France, which are on different topics. INRAE lead 5 Carnot on the 5 big subjects of the Institute: feed, food, environment, and valorization of biomass. And more largely, there is 7 Carnot, which is dedicated to bioeconomy. So I just take a few words more specifically about 3B Core. So, like I said, we are working on the 3 big thematic of bioeconomy: the bioenergies, bio-based molecules, and bio-based materials. And we are, the 18 laboratory and technical center are working on all the chain of processes to begin with the crop production and until fractionation, functionalization, formulation, and they use the green chemistry or biotechnology in a way to make a sustainably production. So I'm available for B2B meeting or talking with you on the chat on the platform if you need more information. So thank you, and the real workshop could begin now.
Okay, thank you. Thank you, Coraline. So now I am going to make a very brief introduction of INRAE. INRAE is France's National Research Institute for Agriculture Food and Environment. It was created on January 1st this year by the merger of two important research French institutes, INRAE and INRAE. As a scientist, our job is to produce and disseminate knowledge to help solve major societal challenges, and to put this knowledge to work for foster innovation, provide expertise, and lend support to public policies. To achieve our goals, we perform high-quality research on numerous topics, including water resources, regional agricultural strategies, bioeconomy, the preservation and restoration of biodiversity, and digital agriculture. We are organized in 14 divisions, of which Transform, which is the division of science for food, bioproducts, and waste engineering. Our major activities focus on structure function studies of biological raw materials and bio-based products, including food and non-food products, and the design and study of processes involved in their transformation. We have considerable strength in the area of bioprocesses, especially in the study of enzyme and microbial bioconversion. We have also competencies and facilities for conducting studies from fundamental research to technology transfer and development. And also we have a lot of platforms for experimental, analytical, or technical studies. So before our speakers start the presentation, I would like to make a brief remain of some important facts about the properties of phenolic acids. Why do we interest in phenolic acids? What makes them a natural versatile molecules with a wide range of applications? Phenolic acids are a subclass of phenolics. They are secondary metabolites with a variety of functions, including plant growth, development, and defense, and they are widely distributed in plants. They are considered as very effective free radical scavengers because of the phenolic group that is available in their structure. The phenolic acid found in plant cells, in plant cell walls and lignin have a unique chemical structure of C6, C3. They are classified in two main groups, hydrobenzoic and hydrocinamic, gathering diverse phenolic acid that you can discover in the screen. Today, I am not going into detail of chemical or of phenolic acid chemistry because we don't have the time. This is not the goal of our webinar. But if you have any questions about phenolic acid chemistry or applications, please let me know, and I will be very happy to send your question to our 3Becar network experts. What gives the bioactivity properties to these molecules are the hydroxyl groups. The phenolic acids are very widely distributed in plants. You can see here some examples of phenolic acid and the plants that we can find them. As you know, the distribution of phenolic acid in plants is specific dependent, but it also depends on a number of factors like climatic conditions, cultivars, etc., etc. So, after this presentation, I give the floor to the moderators for the presentation session. Hélène and Jackie, are you ready?
Yes. Okay, so I can take the floor. Good morning, everyone. I'm very pleased to chair this session, this first session about sources, properties, and potential applications of phenolic acids. We will have, maybe Diana, you can share your presentation, still just to have maybe the agenda of the presentation, if you can, otherwise. So we will have 3 speakers for this first session.
Sorry, yes, I wait.
Okay, perfect, thank you. Thank you very much. We have 3 presentations covering all the supply chain of production of bio-based compound, including first the sourcing of these phenolic acids until up to the potential application and some trends also for the future, and including extraction, fractionation, and purification. So I will briefly introduce, maybe first just 2 or 3 words about what is my research in chemical engineering laboratory. We have a transverse axis dealing with a biorefinery. We are building this axis actually and moving forward for this, strengthen this transversal research in our lab. And personally, I'm involved in extraction, purification using membrane, just to situate a bit my research in this frame. So I'm happy to introduce the first, and also an additional information maybe about the time, average time for each speaker, that is about 10 minutes. And then we will gather the question for the 3 speakers at the end of the 3 presentation with a 30 to 25, 30 minutes time slot for discussion. So I'm happy to introduce now the first speaker, Luc Saunier, holds a PhD in plant biology from the University of Toulouse. As a Toulouse person, I was happy to see that just for the story. And he's research director in INRAE, so that was presented just before by Diana in the Transform department. He's deputy director of research unit, BAA, in Nantes, and research activity focused on several cell wall polysaccharide structure in relation with functional properties in food technology and nutritional impacts in animal and human beings. So now I give you the floor, Luc Saunier, if you want to maybe share your screen and start the presentation. And thank you for participating to this seminar. Thank you.
Oh, okay. I will shut down my camera and share my screen. Okay. So is it okay? Oh no, not yet.
Yes, yes, yes.
It's
yes,
okay.
It's okay.
I guess. So hello everyone. So in this short presentation, I will talk on the occurrence of ferric acid in plant and on its role, but not too much in general, but with a focus on plant cell walls, and you will understand why in a few slides. Sorry. So as okay, what is okay. As already pointed out by Diana, ferric acid is a member of class of compounds widely distributed in plants and called hydroxycinamic acids or phenolic acids. Actually, ferric acid differs from paracomic acid or synapic acid, as you see on this slide, just by the presence or the absence of a methoxyl group. So all these compounds are not found free in the plants, but associated to different molecules, generally by ester linkage. This is the case with glucose and organic acids, with phytosterols, as in gamma-oryzanol, which is found in raspberry oil. Phenolic acids are also esterified to suberin and cutins, which are complex polyesters composing the cuticle covering the outer cells of plants. But ferric acids are also associated with cell wall components and especially with polysaccharides and lignin. So in the next slide, when you look at the amount of ferric acid found in different plants, you observe very large variation, as you can see here, from orange to maize grain pericarp, which is a specific tissue of the grain. Actually, in plants with a low content in ferric acid, this compound is typically found as a glycoside ester. And in plants rich in ferric acid is associated with cell wall components. So note that when I say rich, it is at the base 3 to 4% by weight of the material of the plant, and the pericarp of corn grain has the highest level of ferric acid in the whole plant kingdom. Or this list is not exhaustive, but you can notice that there is mostly cereals indicated in this table, and this is on purpose because the presence of ferric acid is a specificity of grass cell walls, and cereals are part of grasses. But of course, there is some exceptions, and ferric acid can be associated to cell walls in other plants, and this is the case of sugar beet, which is part of these exceptions. So on my next slide, I will explain shortly how ferric acid is associated to cell wall polysaccharides. And firstly, I will just introduce you how it's associated to the polysaccharides of grass and cereals. In these plants, the cell walls are mainly made of cellulose and arabinoxylans. And the ferric acid is linked, which is here indicated as this red hexagon, is linked to arabinose by an ester linkage. In other plants, ferric acid is associated to cell wall saccharides to the arabinine and galactose side chain of pectins, and it is the case found in sugar beet, beetroot, spinach, or quinoa. So we will turn a little bit to the role of ferric acid in the plant, in the cell walls. So an important role of ferric acid is its ability to connect cell wall components together, and it is due to its very high chemical reactivity. This is also why this is a very interesting chemical compound. Actually, it's very easy to form radical species from ferric acid using oxidizing conditions. These radicals are very reactive molecules which like to combine neighboring radicals, and when these radicals is another ferric acid, it's forming a dimer. Well, this is very small on the slide, but there is a variety of possible dimers that can be formed. And that's exactly what is occurring in the cell wall, especially in grass, where arabinoxyline chains carrying ferric acid are bridged together by ferric acid dimers. So the two images, strange looks, it's shells actually, obtained with ferroylated arabinoxylans bearing different amount of ferric acid. The soft shell was obtained with an arabinoxylan containing a lower amount of ferric acid, while the firm gel was obtained with AX containing more ferric acid, allowing the formation of a higher amount of diferric bridge and a stronger network. So that's exactly what's happened in the cell wall. And here, when you look at the content, you see that there is quite a large amount of dimers. There is some variation, but the amount of dimers in the cell wall is quite substantial. So in addition to dimers, trimers and tetramers of ferric acid have been also identified, and especially in the cell wall of grasses. So ferric acid is present in the cell wall as monomer, esterified to arabinose, or as dimers, still esterifying arabinose, but bridging polysaccharides together. Now, the picture is a bit more complex in lignified tissues. So lignified tissues or lignification occurs in plant tissues that have ceased their growth and that need mechanical resistance. This is typically the case of stems, the organ stems in plants, which needs this mechanical resistance. So I will not describe lignin because it's very, very complex, but I just remind you that lignin results from the polymerization of three monolignals that are actually derived from the corresponding phenolic acid. So paracoumaryl alcohol is derived from paracumaric acid, synapryl alcohol from synapic acid, it's very easy, and coniferyl alcohol is not obvious from the name, but it's derived from ferulic acid. And when these three monolignal are polymerized in lignin, they are identified as H, G, and S units. So in grass, arabinoxylans are bearing ferric acid. Here it's indicated in green, but also paracumaric acid esters. This is the blue and the blue hexagons, blue. So specific ether linkage has been identified with ferric acid with coniferyl alcohol. And so in lignified tissue, ferric acid is both esterified and ether-linked, and it's considered as an initiation site for the lignification process. And this lignification process can go on, can be repeated, and you have an establishment of connection between polysaccharides and lignin components through this ferric ester-ether linkage of ferric acid. So what about the paracumaric acid? As I said, it's already esterified to arabinoxylans also through arabinose, but it's mainly actually esterified to the S units of lignin. So I have to say that this linkage, this ester linkage, are quite difficult to measure, and there is quite a few studies which give a complete picture of ferric acid in lignified tissues. So I tried to make, let's say, a summary in this slide, but as you see, it's not totally complete because there is some ND what are not determined. That means this is present, but it has not been determined. As you can see here, it's generally estimated that a very large part of ferric acid monomers and dimers are also engaged or involved in ether linkage with lignin, which means that they are not really available for extraction because they are really incorporated in the lignin components. So the consequence is that generally in lignified tissue, paracumaric acid is the major phenolic acid measured because it's mainly present as ester linked to lignin and arabinoxylans. As you can see, for example, in mestem, the content of paracumaric acid is higher than the content of ferric acid ester. So I tried to present you the key feature of ferric acid in plant cell walls, and my focus was on plant cell wall because in ferric acid rich plants, ferric acid is mainly linked to cell wall components, and it represents up to 99% of the ferric acid is found with associated with cell walls. So this is a specificity of grass and several cell walls, but it is also found in a few dicots as sugar beet, or spinach. Ferric acid is found as monomer, dimers, and higher oligomers linked to polysaccharides. It's also associated to lignified tissue, but I didn't put it in bold because, as I already said, it's esterified and not easily available. So both ferric acid and paracumaric acid are bound to the component of the cell wall. Ferric acid is the major phenolic acid in non-lignified tissues, while paracumaric acid is generally the major component of lignified tissues. And finally, the ferric acid and paracumaric acid esters are easily released by chemical treatment, which can be dilute acids or alkali. But generally, due to its bridging function in the cell wall, it's not very easy to release ferric acid only using enzyme treatments. So I will just finish my presentation with a last slide just to give you a short introduction of my research unit. I will not go in detail because it's a very large unit, but as indicated by its name, our aim is to study biopolymers. It can be polysaccharide, proteins, also cuttings, and to understand their interaction and assemblies, either to build food matrices or to extract biopolymers from plants and generate new materials. So of course, in this unit, there is a team of about 20 people which is dedicated to the study of cell wall components. And also a specificity of BIA is to host an analytical platform with advanced equipment for multi-scale characterization of biopolymers, and a BIPS platform provide analytical service, and you can contact this platform at the address indicated on this slide. So thank you for your attention. I leave the floor.
Yes, please. And
Okay, so
Can
okay.
wait. Sorry.
you can stop sharing your screen so that
Okay.
somebody else can take it. Yes. Okay.
It should be okay.
So thank you very much for this presentation. As I said, we will keep the questions for the discussion after the presentation, but maybe thank you for this taking home message, as you said. And I can maybe keep in mind that ferric acid is relatively abundant in plants, but not so easy, I would say, to extract. And so maybe it could be one of the questions that we can discuss later on. So thank again. I would like to welcome the people who joined us in between, because I see that the number of participants is increasing, and so it's good. So the second speaker is Sébastien Cajot, here of Celabor company. He will talk about extraction, purification, fractionation. That is recognized as very important too. So just a few words to introduce Sébastien Cajot. He holds a PhD in polymer chemistry from University of Liège. He's actually manager of the food technology extraction department in Celabor, where he is coordinating the activities of the biomass valorization platform. So Sébastien Cajot, I can let you the floor if you're ready,
yes.
maybe. Okay.
Yes, thank you,
and
Hélène.
thanks again for sharing with us your opinion and experience about this.
So do you see my screen, just to be sure?
Yes, it's okay.
That's perfect. So first of all, yes, I want to thank the different moderator for the invitation to this workshop, and also INRAE for this for the organization of this digital event of BIOKET. In my presentation, I would like to illustrate through different case studies innovations. Sorry, I think it's better like this now for the presentation. Perfect. So I would like to introduce you different innovative processes for the extraction, but also for the purification of phenolic compounds from different kind of biomass, and also the potential of scaling. Just maybe before getting to the heart of the matter, I'd like to present you the expertise of Celabor and its biomass valorization platforms. So Celabor is an SME that provides technological services to companies in the field of food, extracts, materials, and environment. Through B2B project with industrial, but also through the participation to European initiatives. So I'm the manager of the extraction department, and we develop green extraction and purification processes. We also study the valorization of agri-food byproducts, but also lignocellulosic resources, algae, microalgae refining, and also building blocks recovery from microbial products. We have also material departments that studies development of a new process for the nanocellulose production, but also the surface functionalization. Just to illustrate our technological platform that is unique in Wallonia, so in Belgium, and gathers at lab and also pilot scale different devices for the biomass processing. So we have some example of extraction devices that we have for maceration, but also innovative technologies such as subcritical water, also CO2 supercritical. Also the possibility to make assistance to extraction with the use of microwave, ultrasounds, also pulse electric fields. Post-treatments with centrifuge, evaporators, membrane filtration, dryers. And finally, we have also all a platform in order to study the purification of secondary metabolites, but also in analytical labs in order to study or extract. So the first technique that I would like to present today is the use of high pressure assisted extraction, and especially the use of subcritical water for the extraction of phenolic compounds. So subcritical water conditions are characterized by a temperature that is higher than 100 degrees, that is maintained liquid due to the use of a pressure of tens of bars. And under these conditions, water has a polarity which is similar to acetone or also ethanol, and is indicated for the extraction of less polar compounds such as phenolic compounds. So under subcritical conditions, water is also more acidic and can initiate some hydrolysis reactions, which is really interesting for the hemicellulose hydrolysis from lignocellulosic materials. But of course, we have to mention also some limitation about these technologies, such as the availability of industrial installations, but also the possible degradation of thermolabile compounds due to the use of high temperatures. So at Salabo, we have two scales in order to implement these technologies. So we have a lab scale with accelerated solvent extractor that we can use, but also a pilot equipment that we developed with reactors of around 5 liters that is also able to work with different solvent than water, but always under pressure. We have also the possibility to work with different cycles in order to really try to recover a maximum of phenolic compounds from specific materials. So in this first case study, I would like to present you the extraction of polyphenols compound from agriculture's bioproducts. And we compare, in fact, subcritical water with conventional techniques that use acetone, also ethanol at 80 degrees in order to do this extraction, and also hot water. So the 4 byproducts that we study was apple pomace, barley straw, corn straw, and also oat hulls. And as you can observe, and especially for barley straw and oat hulls, you obtain really a significant increase of the polyphenol contents in our final extract due to the use of subcritical water extraction. In the second case studies, I would like to demonstrate the effect of applying a pretreatment of the biomass by the use of pulse electric fields as a pretreatment before the extraction processes. So subcritical water extraction was used as extraction techniques for the phenolic compounds obtained from miscanthus. So as you can observe, only a short time are required in order to extract polyphenols, so around 50 minutes. And we have compared the extraction between with the use or not of pulse electric fields. As you can observe in the table, we can obtain a really high increase of the extraction yields with the use of pulse electric field for factors of 3. And also, if you have a look on the different quantification of specific phenolic compounds, we have also an increase of these compounds, such as proteocatechuic acid, coumaric acid, and also ferulic acid that are in really high concentrate amount in the phenol extract. So the great interest of using pulse electric fields is the availability of the technologies at industrial scale, as they are already used, for example, in the food industry, for example, soften the shells of foodstuffs such as potatoes. And all these results were obtained in the framework of a national project that is running for them at Salabo, which is Biomat. In this third case study, I would like to show you first result that we obtained in the framework of a running project. It's an entire project, aggregate value, in which the goal of the project is the development of a cascading approach by the valorization of viticultures, but also arboriculture's tree residues into biomolecules, biosolvent, biogas, and fertilizers. So in the present example, we study the 3 different extraction techniques on PA3 residues. And we test, in fact, ethanol at 70% at solvent, and we test it under pressure in the first exam, as a first technique, but also in comparison with maceration and also the use of ultrasound in order to compare these techniques. So it's not easy to read on the slide, but we obtain really higher extraction yield and also phenolic compounds with the use of a pressure and also a reduction of the extraction time. But it was really interesting also to study the use of ultrasound, as we are allowed to work at room temperatures in order to obtain really high content of polyphenols and higher than in classic maceration and also in shorter time. So just a few words about the upscaling. So ultrasound, different sizes are available at Celabor in order to study the upscaling of these technologies, but it is also available at industrial levels. At Celabor, we are able to work until a size of 65 liters due to maceration vessels in which we can add some sonotropes. Or also study the use of recirculation cellule in order to concentrate, make an intensification of the process by a reduction of the volume of the effect. It is also possible to have the same equipment at a larger scale and also for the treatment of thousands of liters at industrial scale. Then in the last case that I would like to present you, it's also the possibility to do a fractionation, but also a purification of the phenolic compounds from different natural resources. So by the use of resin, but also by chromatographic techniques, it is possible to concentrate and also isolate some pure phenolic compounds. So as it was described in this slide, it's possible by an optimization of the extraction processes to obtain high amount of a crude extract from around 2 to 20% of active in general. Then, due to the use of techniques such as precipitation, also resin, it is possible to obtain a crude compounds until maybe 60%. And then by additional techniques such as chromatographic techniques, flash chromatography, PrEP-HPLC, or also CPC, it is possible to reach higher level until 99%. But of course, all this operation have a cost, and it represents about 80% of the global cost of the process. So I think it really must be considered according to the final application of the compound that we would like to isolate. And to illustrate this technology of purification by CPC, so by centrifugal partition chromatography, I just have an illustration. So for confidential issues, I cannot give you the name of the local bark and the specific polyphenols that we managed to extract, but it's for cosmetic applications. So by the use of subcuticle water extraction, we were able to isolate an extract in the compound, so 40% of our compounds in the final extract, which demonstrate really high antioxidant properties. And by the use of CPC, so centrifugal partition chromatography, so it's a liquid-liquid separation techniques that use a rotor and centrifuge force in order to maintain one liquid static. We were able to isolate our targeted molecule at really high purity level and higher than 99%. And so we also compare the activity of these compounds, which was as this last compounds have very high antioxidant activity. So just maybe to conclude, so Celabor as a technological research center search for new environmental friendly extraction solutions. So by for example, using water and replacement of other solvents such as to replace ethanol, for example, but of course, sometimes we have to use higher temperatures. We're also searching for a technology that can reduce the time of extraction, as it was demonstrated by subcuticle water extraction, but also by the use of ultrasound. And also trying to improve the yield of extraction by these innovative technologies. An important point that I tried to demonstrate today is that the use of also of a pulse electric field, or also subcritical water conditions gives opportunity maybe to have a better accessibility to these phenolic acids, and also from recalcitrant biomasses. And I think it's a good opportunity maybe to open the panel of biomass that is available for this kind of compounds. But of course, we have to take into specific attention to the evaluation of the socioeconomical impacts of these technologies in order to study the viability of these new processes, and also take into account investment cost and also specific aspects such as novel food legislation if food is concerned. So it was my last slide, and just thank you very much for your attention, and also specific thanks to all the extraction teams that support me daily, day to day. Thank you very much.
Thank you very much, Sébastien, for this presentation, and to have shared with us again your view, your point of view about this question. This will feed the next part maybe of the webinar, that will be with a roundtable about more discussion about market push, pull or technology push. And I see that there are many questions. I will keep them for after the third presentation, as we explained before. But just a word to say that you can also participants ask questions directly to the speakers if it's very precise questions maybe. So we will see at the end how to manage all this for every one of you who will ask a question to have an answer, right? So now I propose to move to the third and last presentation of this first part. This presentation will move forward in the supply chain, not until the end maybe, but closer to the end at least. It will be about potential application and promising trends of phenolic acids, and it will be given by Professor Florent Allais, who is a PhD of University of Florida and actually full professor in AgroParisTech and adjunct professor of Monash University in Australia. He has built a multidisciplinary research team gathering scientists with background and expertise in life sciences, downstream processes, and material polymer sciences to work on the bioconversion of biomass to high value added chemicals or materials. And so I give you the floor, Florent, if you want to move to share your screen. And thank you again for being here today to.
Of course, yeah. So
experience.
All right. Can you hear me well? And can you see the slides?
Yes.
Okay.
Yes.
So hi, everyone. First of all, I'd like to thank you for the kind introduction, but also to BioKet for giving me the opportunity today to give you some information of the potential, the potential application of phenolic acid. So I've been asked to start this presentation with a few words about our team, the ABI. So ABI is a research and development unit from AgroParisTech dedicated to biomass valorization. So the lab has been created in the late 2012, and we started our activities in February 2013 in a temporary location, which was a middle school in Reims. And then in April 2016, we moved to the CBB, the European Center of Biotechnology and Bioeconomy, with our colleagues from Central Supélec, URCA, and Neoma Business School to work on very ambitious biomass valorization projects. So to be short, our missions are to identify and valorize agro-resources, combine fundamental and applied research to develop integrated process to transform these biomasses into end products. And the idea is to foster innovation and transfer this innovation to industry to create new jobs. And of course, being attached to AgroParisTech, we also have some teaching missions along our research. So here you have a very schematic picture of what we do in the lab. So basically, we start from biomasses such as lignocellulosic biomass, oleaginous, as well as vine. And then first, using fermentation, chemical, enzymatic extraction, we separate the different components of the biomass, such as lignin, semicelluloses, lignan, cellulose, and so on. And then through the combination of green chemistry, fermentation, and separation process, we can transform these biomass components into a large variety of application and products, such as bio-based polymers, antioxidants, anti-UV, bisphenol A substitutes, flavor, drugs, intermediates, surfactants, and so on. So among the different biomass and the different products we've been exploring in ABI right now is ferric acid, and that's why I've been asked to provide you with applications that are currently industrialized or can be eventually industrialized if we can overcome some of the few limitations that have been already discussed, such as price, volume, and so on. So the first thing that I would like to discuss about is the sources and extraction. So Luc told us a few minutes ago that there are many biomass where we can find ferric acid. And we've been working with many colleagues, and there have been colleagues from INRAE, of course, that have been dedicated to the extraction of ferric acid from biomasses. So on the left-hand side of the slide, you can see the biomass that have been investigated. So you have wheat bran, sugar beet pulp, maize bran, as well as rice. So there have been a lot of work dedicated to the enzymatic biodegradation and to release the bonded ferric acid in the biomass components. And then there have been a lot of works dealing with the separation and purification of this molecule, such as the one that has been presented by Sebastian. So here you have the logos of the few labs that have been working on this topic. So there is the FAR unit from Reims, the Genial team from Massy, our team, of course, BIA from Nantes, as well as YAT from Montpellier, and BLF from Marseille. So there are a bunch of labs, not only in France, but also worldwide, that have a lot of interest in optimizing this kind of extraction, degradation and extraction methods. One of the main limitations of this approach is that not only you have to treat a huge amount of biomass to extract a few kilos or sometimes tons of ferric acid, and that means that you have a lot of also of byproduct to deal with after the extraction. And also the cost of such an approach is quite expensive. So if you want to reduce ferric acid for commodity application, that will be a real issue to overcome. So one option that we can think of to overcome this issue is to apply some synthetic biology approaches, such as the one that is shown here. So this is part of a ANR project that we have been working on with Abolis team from Evry and other teams from INRAE, as well as Givaudan Active Beauty. And within this project, our goal is to synthesize ferric acid as well as other para-hydroxycinnamic acid, such as coumaric and synapic acid. And the idea is to start from this phenolic acid to build new antioxidants and anti-UV molecules. So in terms of sourcing here, ferric acid, the goal is to use synthetic biology and engineer microorganisms that will be able to convert first generation or second generation sugars into ferric acid. So there are a lot of work going on. I guess we are not the only one to explore this field, but there's something that is providing us with very interesting results so far. And if you want more information about this project, I invite you to get in touch with Abolis. That will be happy, I guess, to give you more information. So here's some applications. So I want you to start with a very industrially relevant and already commercialized process. And I guess all of you already know about this process, and I guess our colleague from Solvay will be giving more details about it. So there is a current industrial process that consists in extracting ferric acid from rice bran oil and to convert, using biotechnology approach, this ferric acid into vanillin. And of course, vanillin can be used for a lot of application and mainly for food application. And the very interesting aspect of this approach is that since we are using biotechnological process, we can claim the naturalness of this flavor. which is very interesting in terms of price for commercialization. So this is an industrialized process, and there is a lot of market value here. Here you have something that is not commercially available yet. We are still working on the implementation and the scale up of this process, but basically here what I wanted you to show you is that we can use ferric acid in two very interesting fields. The first one is linked to the anti-UV application for cosmetics. So here you have some structure that have been devised and patented in our work. This is still from the INRAE project. So basically, we have been able to, starting from bio-based phenolics deriving from ferric acid, to build new anti-UV molecules that are not only bio-based but also biodegradable and non-toxic. And with our colleague from INRAE, we've demonstrated that such compounds are not endocrine disruptors, so which is kind of very interesting. These molecules are also involved in another project. It's a FET open project called Boost Crop, and we have been working on the use of ferric acid as a chemical platform from the synthesis of what we call molecular ethers. So basically, these molecules are able to convert the UV radiation into heat, and then this heat can be used to protect crops from frost, but also to boost the photosynthesis and increase yields. So there are a dual interest in using ferric acid for this kind of markets. From ferric acid, with our colleague from IGPB and INRAE, we have been working since 2013 on this use of ferric acid to synthesize new bisphenol A substitutes. So they are still 100% bio-based, biodegradable, and non-toxic. And here I wanted to show you the analysis that have been performed by our INRAE colleagues. And on the left-hand side top, you have the endocrine disruption activity of bisphenol A and analogs, and you can see that when you increase the concentration of this molecule, you do increase the endocrine disruption. And in the bottom and side, you can see that for our molecules, whatever the concentration, we don't have any activity whatsoever, which proves that these bisphenol A substitute deriving from ferric acid are very interesting and quite promising. So to go further with this BPA substitute, we have demonstrated with our colleague from ICGM from Montpellier, but also a colleague from Rensselaer Polytechnic Institute in the US, that we can replace bisphenol A in epoxyamine resins, and that we can access very interesting resins with thermomechanical property that can be complementary to that of bisphenol A. And one main advantage of this approach is not only we have bio-based and non-toxic substitutes, but we also have degradable activity and capacity given to these epoxy resins. We can use also these kind of ferric acid derived bisphenols to make some non-isocyanate polyurethane. So that has been also validated at the lab scale. There is still a lot of work to be done to transfer and to access higher TRL to eventually commercialize this technology, but it's quite fundamental at a fundamental stage yet. Something that is also of interest, this is a work from a colleague from the University of Florida in the US. So from ferric acid, they have been able to develop a family of new polyesters that are bio-based, and they can have properties similar to that of PET that you can find in plastic bottle application, for instance. So here there is also a lot of promising potential of these ferric acid-based polyesters. And still, this is still at the fundamental stage, and there are still a lot of work to be done to eventually envisage the industrialization, commercialization of these new polymers. Still in the polymer field, this is a work from colleagues from Rutgers in the US still. So here we're not talking about commodity polymers, but what we call macromolecular products. So basically the idea is to use ferric acid as a monomer to make biodegradable and biocompatible polymers. And the idea is through the degradation of these polymers in vivo, you can release ferric acid. And since ferric acid has some antioxidants and antimicrobial activity, you can have here what we call a drug delivery system that can not only protect ferric acid from degradation when it gets into the body, but you can also finally target the site of release to optimize the activity of the drugs. So this is something that has been patented, and I don't know yet if it's commercialized or not, but this is very promising also application. So here it is shown with ferric acid, but what can envisage this methodology to be applied for all kinds of different phenolics and flavonoids, for instance. And the last application is something quite new that we have been working on with our colleague ICMP from the CNRS at Thiers and Monash University, as well as the University of France. And we have shown that we can use ferric acid-based additives that when they get mixed with PLA, we can turn PLA into a very elastic and elastomeric material that can be stretched up to 400% elongation. And the very interesting feature of this formulation, as you can see on the picture, is that it's a shape memory polymer. So not only you can stretch PLA, which is quite brittle when pure, but you can also get some shape memory properties which can be used as a biomedical application, for instance. So this is something that has been patented and that is currently under investigation to be used as such a for a biomedical application with our colleague from the University of France and the Reims Hospital. So the take-home message here that, as you know, there is a local plant resource that can be used to release and extract ferric acid. However, there is some issue with the volume and the price of such process and approach. So some solution will be to devise more efficient and cheaper integrated process to extract ferric acid from biomass. I've shown you that we can transform ferric acid into vanillin, but actually you can also do the opposite approach. That means that you can start from vanillin and get ferric acid through sustainable processes that have been recently published by our team. So if one option will be to optimize the degradation of lignins to get cheap vanillin and then to transform vanillin further into ferric acid. So that's one of the way we can explore. And the third and most promising way, to my opinion, in the synthetic biology approach that could eventually lead to higher volume, but mostly lower prices. As you can see, there are many applications. I'm sure I have zapped some of them, but there are many applications in very diverse sectors, and mainly high value added sectors such as cosmetic, food feed, and so on, and also biocontrol, even if I didn't show any slide here. And I think that there are a lot of things to do still with ferric acid, even if it has been explored for decades. And the story just began, and I'm sure there are many other applications that people and scientists can think of. And I'm very optimistic about the potential of ferric acid in high-value sectors. So thanks for your attention, and I'll be happy to answer any question you may have. Thanks.
Yeah, thanks to you, Florent, for this very optimistic conclusion and open to many questions. So this is the last presentation of this first part. We are a little late on schedule, but Diana authorized me to, us to have a discussion until 10:30. So we'll take this 15 minutes. We have many questions, so I apologize in advance not to be able to take all the questions, but for some of them who are a bit specific, or other, you can contact afterwards for sure the speakers just to have an answer, right? If we cannot answer right now. So I will take some questions like, a question that could be maybe answered deeper in the second part of this webinar. But anyway, that is with the innovation effort. So it's for the three speakers. I think this is maybe what is interesting in this question is, how do you expect the evolution of the price of ferulic acid? Do you have an idea, or do you have a comment to make on this? So it is addressed to all of you, in fact.
Florence speaking. Yeah, so the price of ferulic acid highly depends on the purity. So if you're on high grade ferulic acid, that could cost between 100 and 150 euro per kilo. And the lower purity ferulic acid will be way cheaper. But for what I know, I don't have access to industry prices, but current ferulic acid price are between, I would say, yeah, around 50 euro kilo. But I guess my colleague from industry could provide us with more precise prices. But there is a huge link between purity and price.
Okay, thank you very much. Another question, that is, what are the current highest yields, if you know, of ferulic acid based on the initial biomass? So this is linked with the concern with the availability and content of ferulic acid in biomass. As one of you, Florent, I guess, said, there's a huge amount of byproduct because ferulic acid is only a very small part of the dry mass.
I would say it's a few kilo per tons, but not higher. That's why I was emphasizing and insisting on the volume and the price, because to get 1 kilo of ferulic acid pure, you will have to treat a huge amount of biomass. So for instance, let's say that you get 2 kilo of ferulic acid per ton, then you would have to deal with hundreds of kilos of byproduct to further valorize in terms of price and cost.
Okay, so now a question about extraction processes, maybe, that is addressed to Sébastien Cajot. Would you say that supercritical water extraction is more effective than more conventional supercritical CO2, or combination of solvent extraction and CO2 extraction, especially for this kind of solutes?
Yeah, I think concerning polyphenols and phenolic compounds in more general terms, we have a better selectivity of affinity, with supercritical water extraction due to a more similar polarity with supercritical water extraction than CO2, which is more non-polar. And main advantage also is that we only use water and not, for example, a mix between CO2 and also ethanol. So I think it is an easier process if you only use water, even if you have taken into account that at the end you have also to dry the phenol extract if you want to obtain a solid extract at the end.
Okay, thank you. There were also some questions that were answered directly between speaker and another question is, does the pulse electric field technique match with the nitrile process or something like this? So I understand that, could we still say that it's a natural biosource molecules when using pulse electric field?
It's a good question about, yes, it's a huge word for a lot of things, but yes, I think so, because in fact you only use water. You just apply it an electric field in order to disturb the membrane of the cell and try to release more easily the compounds. But yes, it's not a natural process. I think it's environmental friendly techniques, but yes, it's difficult to say if yes or not it is a natural techniques or not.
Yeah, I mean,
depending also of all the regulations, because if we are speaking about Cosmos regulation or FDA regulations, they will not have the same answer for using the label natural.
Yeah, of course.
So there is a prolongation of this question that is asked, but maybe it will be too long to answer, like which way technology could be considered as a clean give a clean neutral label. Maybe it can be discussed later on, because I think it's a very large question and we just have a few minutes maybe. Okay, so we can continue at another moment. Maybe I would ask a question for the first speaker, Luc Saunier, because I don't see any questions specifically for him. So I would like, there are so many, I guess.
There are some questions regarding the viability between ratio ester and ester bonds, but also what is the best analytical method for phenolic acids?
Yeah, yes, but okay. You made an answer, in fact, in private. That is why I didn't take the question, but it can interest everybody. Yeah, yes, you're right.
Yeah. Sorry.
What's about the best analytical method to analyze phenolic acid?
Yeah.
Yes.
I'm sorry. I made mistakes with. I believe
good.
It was a general response. Okay, but the best method, of course, you know, this UV, this phenolic acid absorbed in UV, so you can always make some UV absorption method, but it's not very specific because there is a lot of interference that are possible. So generally, they are measured after they are hydrolysis by alkaline. So you have to free them from either from glycoside or from the cell wall by an alkaline treatment, the ester linked, and then you separate the phenolic compounds by HPLC and make UV detection. So somehow you need the standards. It's easy for phenolic acid, the paracoumaric acid. For the dimers, it's more tricky. And of course, for ester linked, it's just, let's say, a pain to measure it.
Okay. But anyway, this concern of analysis is very pertinent in biomass valorization, because you have such a huge spectrum of molecules close to each other that it's difficult maybe sometimes. So maybe one participant or more than one have some analytical experience to share with the others, or reference of publication maybe to give. So maybe you can do it besides the meeting itself. I don't know. Okay, so I don't know. There are some additional questions, of course, but like the sourcing of ferulic acid, someone says today most ferulic acid comes from China. This is at this period a very high concern, more maybe than before. What do you know? What kind of extraction methods do the Chinese people use for this ferulic acid extraction?
Yes, they extracted from the rice bran oil, so which is a byproduct of rice, let's say, industry, and it's very diluted, but it's 3%, 2-3% of the rice bran. The content in ferulic acid is 2-3% of the rice bran oil.
Okay.
Is it a chemical extraction?
What I would say, it's just what you mean chemical.
They use sodium hydroxide.
Yeah.
Okay.
They usually use sodium hydroxide or potassium hydroxide, and then they perform chromatography separation.
Okay, so so maybe just to go further to this aspect, maybe to close, that there would be some challenges to face for the extraction to be improved or more efficient, from cost, environmental concern, or whatever. Maybe Sébastien Cajot, you can have, no?
Yes.
With your point of view about this, because you are working on extraction. I mean, you are not the only one, but the one who was invited today. So,
of course, I think it's we will try to promote the use of that kind of innovative extraction, and because we observe that it is possible to increase extraction yields or have a diminution of the extraction time or thing like this. But it's always there's some restraint, due to the pilot availabilities of this new technology. So I think and also there is some question about also regulation, novel food application of this kind of techniques. So I think it's really interesting that we managed maybe in Europe to have really a specific work in order to try to have equipment at industrial scale available in order to maybe I don't know how to say it in English, to débloquer, to unlock this problematic and try to have innovative technologies available. Yeah.
Okay, so I think, except if you have the other question, a more detailed one, and you can maybe discuss later on or during the second session. So if you agree, we can maybe close this first session and move to the second one. So I give you back the floor, Diana. Thank you again for all of you. And okay.
Thank you. Thank you, Hélène and Jackie. So we can continue now after seeing some examples of what researchers are doing with phenolic acid. I remind you that we took the example of ferulic acid because you are going to see is one of the phenolic acids that have the most well-established applications in the industry. But of course, this is not the only. So we are all interested in developing bio-based or plant-based products to substitute fossil-based materials, and also to bring new functionalities to the market. So plant-based chemistry represents an industrial force that is recognized as a driving element for the bioeconomy. And so in Europe, we have everything to succeed in developing plant-based chemistry. I'm referring to plant-based chemistry, as following the definition of the French Chimie du Végétal Association, which concerns all the chemistry based on biomass resources. Not, of course, I'm not speaking about food products. So this is the definition I am using for plant-based chemistry. So concerning resources in Europe, we had a huge potential for biomass production with a very well-organized agri-food sector. We have highly educated and skilled population, excellent R&I facilities and universities, and also funding initiatives for research such as H2020. But also concerning industrial facilities, chemical industry in Europe is well organized. Referring to logistics with excellent transport networks, railway, pipeline, etc. So that's why in Europe we have already great success story. I share with you this report from the publication office of the European Union. You can see there is a report gathering 15 success stories called From Idea to Market. So, very close to our webinar. So they are examples of different types and sizes of companies, different products and applications, and even with a very large coverage across the Europe, and mostly with European biomass sources. I cannot go into detail concerning this report, but you can find it in internet. But I would like to make only a focus, for example, in carboxyline and this antiscalant product that is made from inulin extracted from chicory roots, which is a very European biomass. So it is possible to make success stories of plant-based chemistry from European biomass and resources. So which is the next success story? Why not a phenolic acid-based solution? You can see in this slide some example of developments made by units of the 3BK network. So before we go to the discussion with our specialists, just a brief some data about the segmentation of the market of the ferulic acid once again, because it's one of the most well established. as Florent said, there is a difference between the origin of the phenolic acid, but also there is a difference between the purity. So maybe this is an approximative market segmentation. It can be, I mean, different, but this is only to keep in mind that there are different aspects or parameters taken in account for the price. So let's move to the roundtable. And before I leave the floor to Jackie, I just want to invite you to think about is there any opportunity to develop a phenolic acid factory using local biomass in Europe? We are going to try to answer to this question during the roundtable. So if you have any ideas, please let me know. We are going to fill in the blanks all together, because this is our goal in this webinar, to determine or to give some key answers for this challenge. So so Jackie, it's okay for you?
Yes, thanks Diana, for this presentation. And just a second, get some problems with my computers. So as explained in the first part of this webinar, we have discussed more about the challenge, the technical and scientific aspect of the valorization of different phenolic and acid, and especially the phenolic acid. And we are putting on the light some proof of concept and potential application like anti-UV, antioxidant, the substitution of bisphenol A, and the development of different polymers like biocompatible polymers. That means we were more in the pushing technology from the bench to the market. And in the second part of this one table, we will speak about the scale-up, but especially we'll try to share a common vision and answering to the question as explained. Is there's a real business opportunity to develop a global value chain on phenolic and a phenolic factory using local biomass in Europe, and for which markets? Will it be for high added value market like cosmetic, like aroma, or mass market like materials, as we can see? And also what will be the key factors, the important key factors, success factors for these factories, and what we will try to fulfill will be again to fulfill what are the drivers, the restraints, and the challenge and the opportunity to develop this this business. So in order to answer to those questions, we have 4 companies in this one table, which will bring us not only their study case, but also their visions. And also we make a small comparison with the existing product on the market, and especially coming from phenolic from China. So you can perhaps show the slide with the different sessions with the different speakers, Diana.
Sorry.
So we will have as 4 companies, Anne Lomas-Colo, which is scientific advisors and co-founder of Oleoinov, and also Elise Odino, technical manager of Oleoinov. Also in the panel list, we got Florence Lutin, which is the scientific director R&D of Eurodier Industrie, which is a well-known advanced separation technology company. Stéphane Bernier from Solvay, which is the R&D director of the global business unit Aroma Performance, and Christian Gombert from the Biotech Marine, which is affiliated to the well-known ingredients inspire CEPI group. So we will first of all give the floor to each panelist and take one or 2 specific questions at the end of each presentation. So I remind you that you can use the question and answer button in order to make some question. We will relate to the speakers, but also some comments and your analysis on the market trends, barrier, challenge, in order to fulfill in direct life the matrix that will show at the end. But perhaps, Diana, you can perhaps show the matrix, and I will try to fulfill a little bit some before launching the first, I will say, the first speakers, just some important point which has been shown. You can show the matrix.
Sorry.
Well,
yes.
I can.
That's okay.
Okay, I can just ask you eventually what has been say until now. So as drivers, we have noted that someone is speaking about naturals. That means there is certainly a growing demand for a natural process or eco-friendly process. So you can write it. Also as a challenge, for example, what has been mentioned until now, that it was very diluted molecules, and we speak about 1 to 4% of to extract. That means also a very challenge of extraction process for specific biomass, and also the viability as a challenge. And we heard also that's necessary perhaps to preserve some properties as a challenge. So for challenge, you can say very diluted molecules, extraction process to adapt to specific biomass.
Okay, sorry, can you repeat? I was spreading.
And viability. And the opportunities, someone mentioned that it's quite abundant. It's something we will look. But I will send you to the some information, and we will try to keep on writing it in direct life. And we need all the participants to help us to fulfill this this matrix. So the first speakers will be 2 speakers, which will be Anne Lomas-Colo and also Elise Odino, which will present OleoInnov. They are co-founders of this new startup, which is developing a patented bioconversion process from synapine to cannolol, which can be used in very various, I would say, sectors: nutraceutical, cosmetic, and Greek chemistry. So you can present in 10 minutes your activities.
Yes, thank you. You can hear me?
Yes, we can.
Okay, so I am going to share my presentation. Okay. It's okay?
It's okay. We can completely see it.
Okay, so thank you very much for inviting us to this workshop. So I am Anne speaking, but Elise is close to me and will be able to answer some questions. So here we want to give you an example of a story from research to market via the experience of our startup called OleoInnov. The idea of OleoInnov was to use the oilseed co-products, namely rapeseed and sunflower meals, as a sustainable source for the production of bio-based compounds, and especially phenolic acids and derivatives. OleoInnov is the result of 6 years of research between the Joint Research Unit of Biodiversity and Biotechnology of fungi from INRAE and Aix-Marseille University, Terres Innovia and Terres Univia. The initial idea of the research project was to develop sustainable biotechnological processes to give added value to the meals. And we have successfully developed such a process based on the extraction and conversion of synapic acid. So you have understood that I am going to speak about synapic acid and not phenolic acid. And the startup Oleoiner was created in January 2019 to scale up and to market the process. Alors, next. Okay, sorry. So, here you can have a quick look to the Oleoiner website if you will be interested in, and you can see the original founding team. Frédéric Finn from Terinoga, who is currently the deputy CEO, Elise Odino, who is the technical director, and myself and the late Professor Seguayo from the BBF lab as scientific advisors. So rapeseed and sunflower meals are the solid part recovered after the extraction of oil from seeds. And in the case of rapeseed meal in France, about 2.2 million tons is produced each year, which makes their economic development essential. As these meals are rich in proteins, they are currently partially used to complement livestock feed ration, but with a relatively low price of 300 euros per ton, which represents a low added value for the sector. But all seed meals contain also phenolic compounds between 1 to 4%. In the case of rapeseed meal, the main phenolic compound is synapine, the cholinester of synapic acid, which could represent a potential of around 20,000 tons per year. So in our process, we use the enzymatic potentialities of filamentous fungi and their enzymes to extract from meals phenolic compounds of added value for applications in industry. But concomitantly, the residual meal depleted in phenolic compounds can be used as a so-called premium meal with increased nutritional digestibility. So we consider the phenolic acid extracted, but also the future of the residual meal. And so in our process, we develop a two-step bioconversion process of naturally occurring synapic acid from rapeseed meal into cannoli by combining the complementarity potentialities of two filamentous fungi, namely Aspergillus niger and Neolantinus lepidus. So in our cases, no chemical hydrolysis, but an enzymatic hydrolysis and enzymatic bioconversion. So in the first step of the process, the use of the enzyme ferrulyl esterase state A made it possible the release of free synapic acid from the raw meal by hydrolyzing the conjugated forms of synapic acid in the meal, which are mainly synapine and synapoyl glucose. And we had the chance that the yield of hydrolysis was very high, mostly 95%. After specific recovery, purification, and concentration with specific resin, synapic acid could be added in a second step to submerged cultures of the fungus Neolantinus lepidus as the substrate of bioconversion into cannoli by a non-oxidative decarboxylation pathway. Cannoli production reached around 1 gram per liter with a molar yield of bioconversion of about 80%
at
lab scale.
You have shown us value chain.
And so this is the process that was patented
the
and
different,
then
I would say,
licensed
biomass from the rapeseed sunflower to the cannellon market. What will be the next step?
Just if you want, I have added the next slide to show a little bit which is our current situation and our
it's really very interesting to because we use you as a study case, so it's important to know what will be the next steps.
Voilà. So yes, it was if we have supplementary question, and especially the implementation and the improvement of the process today. Of course, I don't talk about looking for fundings, which is one of the, of course, one part of our work. But we are currently working on several points. The first point is to reduce the production costs by several items. For instance, by optimizing technical aspects of the current process for scaling up, that is to say, to transfer from lab scale to pilot and industrial scale. For instance, choosing industrial grade quality of chemicals for growth and reaction media, the mode of sterilization and the mode of bioreactor following, etc. The recycling of resin, so different technical points. In a scientific point of view also, you have seen that we have 2 steps in the process. So 2 steps mean time. And so we are working on the second point also, that is to use directly the enzyme responsible for the non-oxidative decarboxylation pathway, that is a phenolic acid decarboxylase, very characteristic and specific from the fungus Neolantinus lepideus. And we are working on the use of the enzyme instead of the fungus itself in the second step of the process to try to shorten the second step. In terms of cannellol, which is mainly the goal of the process, we are working to characterize cannellol at the regulatory level, and we have already paid for the study about that, performed by the DropChem company that characterized the synapic acid and cannellol in terms of reached standard, natural label, and so on. So we have done that. And we are currently also working on the characterization of the true anti-inflammatory and antioxidant properties of the cannellol produced by our technology to confirm also its non-toxicity. That means that in literature, there are many papers describing the higher antioxidant, etc. properties of cannellol, but we want to verify that with the cannellol produced by our technology. And this study is currently performed by the OncoDesign company.
Okay. Thanks a lot for showing us the next steps. There is some questions regarding the target cost for cannellol today. Is it 30 to 40 euro per kilo or higher?
Yes, and also we have made, you have already made a study about that, because we are written a project for eLab funding. So we have made already, how to say, market study and commercial evaluation. And so currently for cannellol, with the current process at lab scale, we are about between around 60 and 100 euro per gram. And we want to obtain, to reduce the cost to 10 euro per gram.
10 euro per gram, okay. And what is the purity required? Is it 90%?
Alors, in our process, after the second step of bioconversion, the purity of cannoli is about, and after concentration by resin, the purity is about 55%. The residual components are a little bit residual of synapic acid and a little bit of syringic acid, which is a component of the pathway of the conversion of synapic acid into cannolol by fungus. At this step of our work, we didn't take the choice to purify completely to 100% purity cannoli by flash chromatography. It's a choice for the moment to reduce the cost, and that's why we want to verify the properties and the non-toxicity of this cannolol with a little bit of synapic acid and syringic acid, with a purity of about 95%, because we will want to avoid the flash chromatography step if possible.
Okay, thanks for this information. And that's this question about purification will be tackled by now Florence Lutin, which is R&D scientific director in Eurodia Industry,
yes,
and which has a very huge experience in separation technology and on biotech chemistry and also in water reuse. So there is also other questions on enzyme limitation. I will ask you to answer directly to the guy. Okay, Florence, so you can switch.
Yes, thank you. You can see my screen. Is okay?
Yes, it's okay.
So thank you to moderators to invite Eurodia at this workshop, of course, and specifically on what are the scale-up challenges, because on our industrial experience, we have something to say, of course. So first, a brief introduction of Eurodia company. Eurodia is a process engineering company located in Pertuis, close to Aix-en-Provence. And our core business is to propose advanced separation technologies as a membrane filtration system, electrodialysis, and ion exchange technologies for demineralization, fractionation, purification of liquid solutions. So Eurodia is involved in several agro-food markets, mainly in dairy, wine, fruit juice, sugars, and sweeteners. And in biotech, Chemistria is a label division dedicated to this activity. We have also an activity in chemical industry, and more recently in water reuse with electrodialysis, specifically for desalination. Our activity is international activity with a branch office in US, name is Ameridia, branch office in Brazil, EEGB, and in Russia, Eurodia Cuban. The services offered start from R&D until to the construction of complete units. So R&D division takes in charge the feasibility study with lab test and pilot test. The process division defines the full process line and site equipment. And at the end, engineering team makes drawings, PID, and follows the manufacturing of the unit. Eurodia provides to the customer CapEx, but is engaged in process guarantees with OpEx, means including the cost of energy, water, chemical consumption, and membranes replacement. So it's very important, of course, for Eurodia to start from the beginning and for the development of the process line. In green chemistry, Eurodia has a long experience since more than 20 years, specifically for organic acid production. We worked with Hélène Roux at the beginning, with LGC on this thematic, and specifically for lactic acid, large lactic acid production and succinic acid factory, and more recently also in purification of 2G sugars from wood and fin material where phenolic acids can be also considered. And also I follow with INRAE a subject of coffee beans and the recovery of phenolic acid on coffee beans during a PhD with Montpellier INRAE. In fact, to get a pure molecule from biomass or fermentation broth, the sequence of steps is always the same. And cracking of biomass or clarification of fermentation broth, separation liquid solids, followed by purification, extraction, and at the end, concentration. So for such molecules as phenolic acid or other organic acid, we discussed before, first issue, and I just summarize what we the main important items that we have to take care. The first issue can be the low concentration in biomass or in bioreactor. And for this reason, the selected technology must show a high recovery. Second point is impurities level and purity targets. As soon as the impurities considered are high, the sequences of steps becomes longer. At the end, needs to consider the deletion of target molecules, usually because the concentration is low, a lot of energy is needed for concentration of molecule of interest. So if we consider the purification step, membrane technologies are very useful and convenient to recover natural molecules. It is a friendly technology, soft treatment. But if really interesting, if at the end we want to reach only a pool of molecules with a different sequence of different cutoff of membranes from microfiltration up to ultrafiltrations. This technology allows to obtain a pool of molecules with a certain demineralization rate if we add nanofiltration, for example, or electrodialysis. But this is a pool of molecules, but sometimes it's enough. So it's very important to ask the question, what is the level of purity we need to reach? If the level of purity is not enough, other technologies with a higher specific selectivity can be considered, and specifically ion exchange resins, specific adsorbent resins. Mitsubishi is in our chairs holder, so we have several adsorbent resins very suitable for this purification, or ion exclusion chromatography. But so this is a certain purity, but sometimes it's not enough. So, and specifically for pharmaceutical grade. So at this end, we need integrated more selective technologies, has a high pressure chromatography. We spoke also of subcritic water, of subcritic CO2, solvent extraction, crystallization, distillation. So needless to say that at each purity level targeted, the associated of CAPEX and OPEX has considerably increased. And during this process, of course, you generate waste, byproduct. So think about what we have to do with this byproduct. It can be sometimes becomes a co-product, but the question is very also important. So this is really a brief presentation, but I think that the key question before starting a project to market a natural molecule, several questions must be asked and are summarized in this slide. Other than the selling price and the market opportunity, you have to consider the concentration of a targeting molecule in the raw material. And if the concentration is low, of course, look at the feedstock availability. And don't forget that. Low concentration means also large amount of water and impurity to be removed. To select the most appropriate technology, we show a very high yield level and allows to achieve the level of purity set. So at the end, the message of this presentation is don't minimize the downstream process, the complexity of technology that you have to integrate, the OPEX linked to the energy, chemicals, and all other water and so on, and also the generation of waste during this process line. So why we insist on that? Because this is really the challenge. And so perhaps it's very simple for everybody, but when we start a program, a large program or investment, we have experience specifically for organic acid, lactic acid, succinic acid. So a lot of money and energy of study was spent on bio reaction, on fermentation reaction, and very low time at the end and low financial for the DSP. And the success of that is not very good because majority of large plant expected 10 years ago is not now in the market. So this is my message, our message. So thank you for your attention.
Thanks for this which will help us to fulfill our matrix, and especially on be careful about specification impurities and concentration of pool of biomolecules you want to develop. Just a question also about how do you preserve the properties of the biomolecules, which is also quite important, I think, for phenolic acid specifically. What does it mean for you? Is it?
Yeah. I don't mention that, but this is also a key question: how to preserve the activity of molecule, specifically antioxidant molecules.
Yeah.
So during this study with INRAE, we also saw this problematic. It was not phenolic acid. It was what is the molecule was.
Chlorogenic
yes,
something like
this
that.
Is and chlorogenic acid, and
Yeah.
this chlorogenic acid is very affected by oxidation. So in fact, we think to operate in nitrogen atmosphere for certain part of the system, and so but this is more complex.
Yeah, quite complex. And can you mention how much is the cost of the separation compared to the global cost production?
In fact,
give us an idea in percentage.
No, yeah, it is what we have mentioned. And the cost represent usually 80% of the cost of the production, the DSP. Yeah. So depending of the value of the end product, we need to cover this cost and take care. Yeah, the consideration of the investment cost, but the OPEX, and in many cases we need also to add a certain process to recover water, to minimize salt rejection, and so on and so on. So this is also a real a complex approach, and I think that we need absolutely to include this process in the concept of biorefinery, with a possibility to have a byproduct, co-product with a certain value.
Okay, another very important key key success factors is thinking about the global approach and biorefinery in general, and don't care about be very careful about the DSP, which is very important in the development of such product.
What we
okay, thanks,
before,
Florence.
Okay, we have a discussion.
Yeah, thanks, Florence.
Thank you,
Yeah,
thank you, everybody.
We'll
Thank
take
you.
the question after, perhaps.
Okay.
Okay, so we now switch to Solvay, which is already selling some vanillin from ferulic acid from rice. So it's Stéphane Verdier, which is the R&D director of Solvay Aroma Performance Global Bio unit, which will present the vanillin, and we'll ask him is a potential market now for replacing vanillin, natural vanillin.
Sure, Jackie.
Yeah, that's good.
Can you hear me? Can you see my screen?
Yeah, it's very good.
All right.
It's nice.
All right. So first, thanks to the organization committee for offering the opportunity to Solvay to testimony on what we do on natural vanillin from ferulic acid. My name is Stéphane Verdier, and I'm in charge of research innovation for the Global Business Unit Aroma Performance. So I will not introduce the multi-specialty chemical group bearing the name of its founder, Ernest Solvay, more than 150 years after the description is made on soda ash. But just let me tell you a few words about the Global Business Unit Aroma Performance. Basically, we are a SME with global presence, serving 4 markets and being the historical market leader for vanillins. So now moving to the topic of today, Solvay is indeed leading the growing market of global natural vanillin made by fermentation of ferulic acid. At this stage, I think it's interesting to give you some background of natural vanillin and, for instance, what I call as a global natural. As already mentioned this morning, regulation on naturalness, naturality, whatever you call, are not really harmonized. But I think there is a common agreement, at least for the food flavoring ingredient, that the European one is the most demanding today. So if a vanillin is considered as natural in Europe, it shall be considered as natural everywhere. That's what we call global natural, because some of our customers are willing to have an ingredient, a natural ingredient, one fits all, that they can formulate everywhere on the planet. So this is the case today for the natural vanillin made by fermentation of ferulic acid, providing that the ferulic acid is obtained by a non-GMO natural feedstock and is recovered by, let's say, traditional processes. The European regulation is kind of generic, let me say so, but it says, does our ancestors, would they be able to do the same job than at that time? If so, it could be considered as a traditional process. And today, the conversion of ferulic acid into natural vanillin by a fermentation, we all know that alcoholic beverages, fermentation with yeast is operated since millennium. So Solvay pioneered the commercialization of natural vanillin from ferulic acid in the 2000s. And at the time, for the first 15 years of production, it was a very, very niche market and actually not really growing. And only over the past 5 years, the demand really started to grow, really started to materialize due to, from our perspective of Solvay, 2 reasons. First, all of us as end consumers, we are willing to get more natural stuff, natural ingredient, natural something in the food we are all eating. And second, the fact that the vanilla bean extract are not able to serve such a big market. So it led Solvay to launch a new grade in 2018 with high purity, what we call raw vanilla natural CW, with all the requirements from the food and flavoring markets. We increased the capacity. We have made an announcement in 2019 last year. And now it has for sure implication on phenolic acid. Today, this is what is materialized on the left on the graph, on the slide. It says all of it comes from Asia. All of it comes from the rice bran oil. And basically it has been highlighted this morning as well. I think it was Luc or Florent. Basically the co-product generating during the rice bran oil production is used to derive gamma-resinol and then to hydrolyze into phenolic acid. So that's the situation today. And I was telling you that the market was growing. I want to, with this slide, try to highlight what we intend to do at Solvay. I would consider that the current growth we observe is rather incremental. And what we need to achieve is rather a step change. On the left, you have the history, the vanilla bean extract from the Titaensis, the Planifolia, Orchidea. Today, it's the way the history of vanillin started with. But even with the advanced agronomics we can offer today, the supply will remain very limited. And if we express the vanilla bean extract supply in equivalent vanillin, equivalent molecule, today I consider it's 20 tons a year. On the right, you have the 20,000 metric tons of a year for food, flavor, fragrance markets. All of that is almost fully served with synthetic. And unfortunately, there is no way for vanilla bean extract to reach such a 20,000 metric tons a year. In between, that's the reason why in between there has been this natural vanillin ex-phenolic acid market that has been created. I told you that at the beginning in the 2000s, it was 10, 20, 30, maximum 50 tons a year. Today we succeed to grow with all our peers in the industry, and we consider it's 10 times bigger compared to the vanilla bean extract, but still 100 times smaller compared to the synthetic vanillin. So here on this slide, there is a typical example of elastic market illustrated here, because the volumes are driving the price. Let's say the 20 tons a year for vanilla bean extract, it's a price of gold. On the other hand, the 20,000 tons of synthetic vanillin is the price of iron. And in between, you have a kind of copper. I don't know if it's the right pricing, but you have a kind of specialty named copper, and all the intent we have to serve the demand of our customers, the demand of end consumers, is to move the natural vanillin toward the right of the graph, to grow the volumes, but there is no way to make it largely available without decreasing the price of it. So it has consequence on the price of the demand of the phenolic acid, but today the main concern is on the price of the phenolic acid. So that's the situation today, and the survey perspective of what needs to be achieved to meet this demand is that a new natural supply chain shall be developed. Today, we all know the reference supply chain from the oil, very efficient, very mature, well established, very much appropriate to provide what I would call active ingredients for several markets: food, feed, fragrance, home personal care, pharma, materials. But in parallel, I try to illustrate on this slide, the green arrow is much less developed, and there are few supply chain of whatever you call it, natural, biosourced, bio-based, renewable. And that's a pity, because there is plenty of natural feedstock, whatever cereals, oil, sugar the sugar co-products. I think that, like mentioned by Diana, Europe, with a strong agriculture as a card to play. In terms of chemistry, the I try to illustrate with the green beaker is, I think we can manage the natural materials into natural active ingredients or bio-based active ingredients. But what is really missing, that's in between what we call at Solvay biorefineries. So that's where our effort should be put. Two few examples of biorefineries. How can we make it happen to fully crack the natural feedstock into all the valuable molecules there inside, whatever molecules, building block or biopolymers? Our perspective, building on the discussion that started on the competitiveness or price, how can we make it economically attractive, that the full biocracking is kind of mandatory to access competitive ferulic acid price, because as it was several times mentioned, the concentration of ferulic acid in the feedstock is low, a few percent at best. And if only the ferulic acid is bearing the cost of transformation, of extracting the stuff, we will never be able to move down the price of ferulic acid. So today it's not a surprise from our perspective that the rice bran oil biorefinery, it's one of the only few examples I have in mind. I think it's a very inspiring one because these guys develop over probably centuries, and they start valorizing the oil for sure, but also the gum, the wax, the fibers, the fatty acid, the phytosterol, the chlorofenols, oryzanol and ferulic acid. They succeed to valorize almost all what could be valorized in this industry. But I think it's something we should be inspired and we should strive to develop for whatever the corn fibers, the sugar beet pulp, the oat earths, the wheat, because as was explained by Luc, the theoretical potential of ferulic acid is millions of tons. The hard topic is to get at economical and competitive prices. So that's all I wanted to share with you. Before thanking you for your kind attention, just to mention that the natural story at Solvay is a tiny part of the greater whole we call Solvay One Planet, in which we care for products, processes, and now circularity, because we are part of a member now of Ellen MacArthur Foundation. And we try really to move the needle to improve the situation, whatever climate, resources, and better life in general. So thank you for your kind attention and looking forward to your question during the table ronde, the round table.
Okay, thanks for your presentation. So we have got the message, big potential of millions of tons, but we have to think about biorefinery. So you really think there is a premium price for product coming from local biomass, or?
Premium price, not really. I'm convinced that if we make all the good brains, the good technology to fully crack some feedstock, and there are plenty of European feedstock we could valorize, I'm pretty sure if we share the cost of extracting to the different component of the biomass or this feedstock, we could get not premium, but very competitive access to this molecule, biopolymers, and it will be the start of a new supply chain that will feed the different end markets with more renewable or bio-based products. That's my conviction. But you need to take it have a holistic way, not focusing, I do the stuff only for one chemical or one molecule.
And how do you explain that the success in China? How long does it take? So what's the history behind the ferulic acid production in China or in Japan?
They started with the historical usage of rice bran oil because they heat rice. Then from the rice bran, they give some stuff to the cattle for feed. And they found out that if they press the rice bran, they get the oil. And then they develop the use of oil. And then from the byproduct of oil refining, from the crude oil to the edible oil, they generate a lot of byproducts. And each time good common sense, traditional common sense, farmer common sense, they try to valorize each of these byproducts. So it takes a lot of time, and it was a, let's say, a natural, slow, progressive process. Now we have our society are looking to more naturals, more renewable. We have probably 2 centuries of technology behind us. I'm pretty sure we can accelerate what the rice bran industry has probably taken several centuries to build on.
Okay. And do you think that we have to begin first by high-value-added molecules or by, I would say, mass molecules like materials to develop the biorefineries?
I tend to think that the high-value molecule shall be the main driver. But then very soon, or even at the beginning, you need to consider also to valorize the low value. Because if you have 1 kg of phenolic acid and you generate 999 kg of something you valorize at 0, it will not fly. But if you valorize at more than 0, and it can be 0.1 euro per kg, when you sell the phenolic acid at 25 euro per kg, it is the fact that it is non-zero valorization is really key to make the economic attractive. That's our point of view at Solvay.
Okay, thanks. Thanks for.
Jackie, could I ask a short question, please?
Yeah, surely.
No problem. I tried to find the Q&A button, but I don't have access apparently. So maybe because, okay. Stefan, you show something with the complete supply chain from the sourcing of the very raw biomass until the final molecule. Do you have an idea where is the more restricting part of this? Or you understand? So is it more limited by the sourcing or by which step is the more, is a limiting step, in fact, in this?
I think here, Hélène, the biorefinery is the limiting. I mean, the feedstock, we have good farmers, cooperative that are producing a lot of cereals, oil, sugar. They generate a lot of co-products. Today, they consider the co-products are already valorized because they give it to feed. But many of them are aware that if they crack, they could valorize, they could create more value by cracking instead of giving to the feed. On the other hand of the chain, there is a chemical industry like ours, and we say whatever the molecule, is it fossil or natural, we don't care and we can derivatize. That's easy. But the missing part is in between, how to convert the feedstock into the chemicals or the molecule, the biopolymers. And to do that, that's the limiting step, I think.
Thank you very much.
Okay, thanks. Is there other questions and answers?
Anne from BBF, I have a question, but maybe it is a secret, I don't know. The microorganism, the bacterium, I guess, that makes the conversion of ferulic acid to vanillin is not GMO?
Yes, correct. We are operating a wild type microorganism.
Yeah.
Because remember, 20 years ago, it was a GMO, no?
Sorry, Anne, I didn't get your question. 20 years ago, it was?
It was a GMO.
I don't think so, no.
No?
Okay. There are other companies that try to develop a genetically modified microorganism.
Yeah.
But today, the market is served by almost exclusively, to my knowledge, wild type microorganism. Today, the situation in Europe is okay. I would say they could consider vanillin made by a GMM as natural. Now, there is a concern about the customer. We sell the vanillin to customers, companies for the food and beverage industry, and we are in contact with every one of us, the end consumers. And that's true that at least in the Western country, especially in Europe and especially in France, kind of reluctant to anything that starts with G.
Yeah.
In genetics.
No, sorry. Probably I made a mistake with another company because 20 years ago we worked a lot also on the production of vanillin from ferulic acid, our enzymes and fungi. And 20 years ago, from the directive of EU, we have to avoid absolutely a GMO microorganism, but absolutely it was definitively not. And just I was wondering if today this feeling has changed, but you have replied to my question finally.
Okay, thanks. So we'll come back on some question after. So now is that I will share my screen and I will show you. I have tried to make a sum up of all the presentation, and then I will ask all the panelists to react. So I have tried to fulfill this matrix. Noted that there is perhaps growing demands for renewable raw materials, for naturals, and for specifically high value added market like cosmetic, aroma, and fragrance. We have noted that there is a lot of property of bioactivities to preserve on phenolic, and also that the idea is to develop eco-friendly process, as mentioned, CEPIC, natural, with natural label. We speak about natural, about the PEF process, local sourcing, short cycle, and traceability. The challenge we have already shared was that it was very diluted molecules, 1 to 4% to extract and purify. It was quite complicated to develop a bioprocess extraction with wide, I would say, biotech microorganism. But it's something you have to adapt to the specific biomass. You have to take care about viability and purity. We don't speak about sensory things, a little bit on toxicology, perhaps you could add after. We speak about the sensitivity of certain molecules, and we need to preserve their properties. We speak a little bit about the functionalization at the end, about the solubility increasing. Everybody speak about, mentions the necessary of having a biorefinery approach with a full cracking and full valorization, and that means to answer to the different market and to answer to different specifications and requirements. As a restraints, there's all the label with the heterogeneous regulatory kosher, we mentioned also Nagoya. We also speak about existing product on the market that we see with the phenolic acid, especially on beauty care, with some competitors existing, some IP. But there is some opportunities with the market demands. I have mentioned all the figures you gave to me, and so that's certainly a market more than 750 tons per year. Also, there is a few producers of phenolic from China or Japan. So it has already been demonstrated some properties. So perhaps I will ask every panelist to comment this matrix and perhaps to add some elements to this matrix. First of all, perhaps who want to begin?
So wait, Jackie, just a little question. In brown, we can read the words in red, written in red. Difficult for me, for instance, to read the red words. Yes,
Is better like that?
No.
I made
another color.
Maybe.
Yeah.
I would maybe just okay. You change the color. Okay.
I put in white in
white
5 minutes.
Red is
who can begin? So white is good?
Yes, white is good. Yes.
Okay.
Thank you. Thank you. Sorry. Yeah.
No, it doesn't matter. So who want to react first of all? Perhaps Anne Scolo or Elise Odino? A reaction on this matrix? That's good. In full screen.
In fact, some points are exactly what we met when we try to go to the market. That and all the remarks were sent. In our case, since the beginning, we thought about the co-product. That means the residual meal. So it was very important for us to think about not only the high added value molecule extracted, but the residual byproduct. That is for us the residual meal. So I am okay to about that. I think Florence has underlined this point very well. We have to talk to a circular process. So also what to do with the residual biomass. It's very important. And of course, for us, the thing is to reduce the cost. In our case, the problem was not really the sourcing, because in fact, we were contacted by Terrinovia and Terrinovia because the source, the biomass, was here in France, and the problem was not the sourcing, but what to do to add value to this biomass. So for us, the problem is not the sourcing. It's okay. Our feeling is that when we are beginning to think to biorefinery, biotechnological processes with, in our case, enzymes or microorganisms, etc. It takes time. So time is money for the duration of the process. And for me, this time, which is necessarily related to a biotechnological process, seems to be a problem compared to chemical processes that are very rapid, etc., etc. And I think we have to change a little bit all the, how to say, if you want to make green chemistry, biorefinery, and so on, all the chain of the industry, the financial, and so on, have to accept the idea that the protocol are different, and the time of the process is different. So we have to accept that, because if you continue to compare always the cost that are to be very low as a chemical cost, I think it will be very difficult to change our future from all chemistry and fossil chemistry, petrol and carbon, fossil carbon chemistry, to biorefinery and biomass chemistry. Do you see what I mean?
Yeah, I really see how much you develop biorefineries and answering to all requirements and of the market. About your business model, your return of investment or your scaling up, it's for 5 years, 10 years. You know that in biotech, for example, it can take something like 12 years, the last chemical intermediates.
For the moment, it's 5 years, but of course, it's in a reasonable way, rather 10 years, of course.
Okay.
But
okay,
it depends also, because if for canola oil, for me, the interest in canola oil is that for cosmetics or nutraceutical and so on, in fact, in a product, the need of the compound is probably very, very low per gram of product. We need to put a very, very, very low amount of canola. So in fact, maybe interesting in canola for us is that we don't need to make very, very large scaling up and so on to have year production sufficient to satisfy the market. Do you want do you see the idea? That means that we can have the biomass, no problem.
That means it will be not a bigger, okay.
Yes.
Okay, I can see.
Yes.
It will be not a big factory.
It's not so large, so the scaling up is not so large also. And for us, it was one of the reason to go forward to the startup in this for this compound, in fact.
Okay, thanks. So we'll switch to the other panelists. Some reaction regarding this matrix. Florence, or last word regarding the key success factors?
I think that there is a good question regarding Sébastien Cajot mentions innovative technologies, and I think it's very interesting. They mention technologies. In fact, the problem is the scale up of this technology, and it's a technology at certainly volume to treat a certain volume with high only a few liters, 65 liters, something like this, but not more. So also this is a big question. If we probably these technology are very interesting and very eco-friendly, but if we need to move to a larger scale, an industrial scale, we have not the equipment. So why? So this is also a question. Why these all technologies that he mentioned has not now an industrial equipment? So I think also is interesting to mention that if you make a scale up with this kind of machine in laboratory, and at the end you need to produce a larger amount of product, you cannot follow by industrial scale up because there is not the machine, the equipment.
Okay, they didn't react. They can react eventually. Stefan, regarding the use of different technology like PEF and subcritical waters, and thinking about developing something.
I have just a comment. I can make a comment because for us, of course, I always speak for our key markets, but the technology is just a tool. So for our development, whether to get the double or third for 3 times more polyphenol content is not always the key objective. Well, the key objective is to get the best performance or biological efficiency. So when we develop an ingredient, we will test different technology, and we will select the one who provide the best compromise, I will say, between the price, the price, the efficiency, and so on. And that's part of the difficulty today, even for our company, for example, to decide in which technology to invest internally. So today, when we develop a new ingredient, we test different technology, and we will use for one development, we will use one subcontractor. For another one, and sometimes we will produce completely fully the ingredient in our company. So it depends really on the market and the final objective. That's the problem between the green chemistry, when you will look at synthetic alternative, and generally the cost is an issue because we have this problem today in our company. Even if it is much, our customer want natural ingredients. They are not ready to pay for 3 or 5 times more the price. So
Yes.
that's a problem.
And what do you think about using enzyme today? Do you already in CEPIC use enzyme? And what do you think about using enzyme and different technology of extraction for phenolic production?
Yes, for us, so we are not integrated in enzyme technology. We have now dedicated people, but we work with partners. And today we have, even in one specific market like beauty care market, we have 2 approaches. We have one approach for excipient functional ingredient where we accept to use enzyme and even GMM enzyme, of course not GMO. But for active ingredient development, today it's a no-go. So even in one market, we can have different, I would say, market acceptability linked to customer, in fact. And that's the case, for example, if you take the case of nutraceutical, where we are, United States today, in United States, it's very difficult to develop a new nutraceutical ingredients issued from GMM with the positioning of active ingredient, because that's always the question of the ingredient positioning.
Okay, so you need wide microorganism if you want to develop nutraceutical.
Yes.
Using enzyme. Okay. Okay, perhaps Stefan, you can react also.
Yes, that's good, because you have the monthly siren that was working in Lyon. Sorry. So I hope it's better now. I think
it's good.
Great. The SWOT is collecting a lot of valuable insights. At the survey, our CEO has a mantra saying an eye on the microscope and an eye on the telescope. I think what we intend to do, all of us, is to develop an alternative supply chain to oil, to petrol. It will take time for sure, but I'm pretty sure that we can be not probably 10 years, but 20, 30 years down the road, quite competitive and very close to the competitiveness of what we experience today on the oil. And that's good, because in between the oil will have disappeared. So sorry, it restarts. But on that, that's the telescope. Now on the microscope, there are some markets, food is one, fragrance is another, cosmetic maybe a bit less. It could start to subsidize this new supply chain, prime the pump, triggers something, and then I'm sure that we will have the learning curve. You start the biorefinery, you have the learning curve, and so on, you improve. That needs, however, to take a global approach, to check the economics, and make sure that when you start something, the blue sky scenario is attractive. Because if you start something and you know the blue sky scenario it will never be attractive, you better stop everything. That's the point of view. I really think that folic acid, some product for cosmetics or fragrance, can prime the pump on these biorefineries, and then step by step, we will try to expand the valorization of all the stuff we cover. That's all.
Okay, beginning by the fragrance and the cosmetic outlets, and after developing. Okay, other reactions before asking Diana to conclude?
Jackie, I just have a question for our panelists. What do they think about speaking about bio-based chemistry or plant-based chemistry? The question of drop-in versus new molecules or dedicated molecules. Is there because when we hear, I mean, a lot of the developments are made based on drop-in molecules. I mean, looking for a substitute. But what about the new functionalities or new molecules? Is there any opportunity or not? What's your opinion?
You ask to everybody, or?
Yes. If someone wants to answer.
I can a little bit answer because I've worked since 15 years on the development of bio-based products, and you know that we have tried to develop succinic, furan decarboxylic acid, and there's been some success on 1,3-propanediol or on isosorbates. But now with the price of the oil price, that shows that the whole biotech company is now more looking at producing high value-added molecule for cosmetic or for fragrance or for pharmaceutical than really to copy petrochemical building blocks. But there is still some company like Michelin and so which can and which have the time, because it takes time. We have mentioned that, which have the time to develop new building blocks and to develop eventually a process for using not only petrochemical but also biomass at the same time with the same process. So it's possible, but generally what we mentioned is that you have better to copy, better to target some molecule with oxygen, a more added value molecule than pure petrochemical olefin, I would say.
Okay.
If I can just add
now.
Just maybe this is in relation with a concern. Maybe I don't know. I'm not sure that it was listed, but as the question of purity or which purity for which market or something like this, and it's in relation with this biorefinery concept, because maybe for some application you don't need a pure fraction of something, and so maybe can be also. I don't know in this SWOT analysis.
Other points. I also add other points to this matrix. Maybe I can share
I don't know where to put this. In fact, it may be I let you the decision, because I don't know if it's a driver, restraint, or whatever. But I think it's something. I don't think everybody think. I mean that it's also something to be considered, maybe, and that it could be a difference with oil. So
okay, I think we are going. Stefan, you want to say something? Okay, go ahead.
No. I just want to suggest I need to leave. I type a message. Just
okay.
You all. Sorry. Please have a nice discussion. Sorry, guys. Bye.
Thank you. So I think it's time to conclude this webinar. Thank you very much to all the participants. Very few words. I think, like Florence says, the story begins. I think you can see in this matrix that there are a lot of drivers and challenges and opportunities and restraints that we need to work on. I also add in the restraints the question of the stability of the molecules that was highlighted by Christian Gombert. How to keep this properties. So I think there is also work to do on that to stabilize by functionalism, or I don't know, these molecules. So I will just like to thank all our participants. And what I propose to you is to keep in touch if you are interested in these topics. In the 3 Bécard network, you can find the presentation and the result of our matrix in the 3 Bécard network website. We are going to complete, and please continue to discuss and to exchange with us if you have any ideas or questions. We are, of course, open to you, available to answer. So I think that's all from my side. Maybe someone wants to say anything?
No, thank you. I think it was very interesting workshop, and with different people coming from industry, research, and the market, and so the roundtable was very, very interesting, I think. And at the end, we have a support which also have the driving force to develop this study and to start. As we mentioned, it takes time, and it's very complex. Biotech is complex, but natural molecules has also a very important market in future. So we need to work on that, of course. But this roundtable was, I think, very interesting. Thank you to invite us.
Thank you. Thank you.
We have all the elements today to think about making a demo plant.
Yeah.
We have all the value chain. We have analyzed all the drivers, the potential markets, the competitors. And now I'll say we have to create a project.
Yeah, it's very effective.
At the end of the webinar. So that means if someone which is still hearing us, if you are interested in making a demo, or you are looking for, you want to look at other outlets of the products or the properties, so do not hesitate to send us some message in order to develop this demo in Europe with local biomass.
Okay. Thank you.
Thank you.
Bye-bye.
Thank you.
Bye. Thank you very much. I think there's some. Okay, you just no question. Okay. Thank you very much. We remain at your disposal if you have any questions or if you have any ideas to share with us. Thank you.
Bye.
Thank you. Bye.
Thank you very much.